Okay, disease and epidemiology. So what is epidemiology? Well, it's the study of the geographical distribution of disease and the timing of that disease, and how diseases are transmitted and maintained within populations. So it's really a study of diseases in populations. The goal of epidemiology is recognizing and controlling outbreaks. Epidemiologists focus on the etiology, or the cause of the disease, and the transmission, or how diseases are spread. So epidemiologists really focus on analyzing diseases in populations. A population is a group of individuals who are considered at risk for a disease or a condition. It's usually geographical. It can be associated with certain behaviors like IV drug use, or owning particular pets, or living or working at certain places, those kinds of things. And when we talk about disease, we talk about morbidity and mortality. Morbidity is the state of being diseased. You're sick, you have a disease. The morbidity or total morbidity are the numbers of individuals that have the disease. The morbidity rate is the either number of diseased individuals out of a standard number in a population, or like a percentage of a population. And mortality obviously is death. So total morbidity and mortality, here is a very dated example now. As of April 9th, 2020, there were 427,460 cases of COVID in the USA and 14,696 deaths. So that was the mortality. The total number of cases is the morbidity. Now that number is much, much greater now. Prevalence is defined as the number or proportion of individuals with an illness in a population at a given time. Okay, so how many people in the USA have the disease today? Incidence is the number of people who got the disease or were diagnosed with the disease in a certain period of time. So incidence is how many people are diagnosed with it in a certain period of time. Prevalence is just in general how many people have it. So those are two very important terms to be aware of, and be aware of the difference between them. So let's talk about patterns of incidence. So some diseases are sporadic, some are endemic. A sporadic disease is seen only occasionally and without a certain geographic concentration. So rabies, plague, tetanus, they're very sporadic diseases. An endemic disease is constantly present in a population or specific geographic location. Malaria, Ebola are great examples. Epidemics are diseases that are occurring a lot more than expected in a short time within a geographic region, like influenza has epidemics almost yearly, right? A pandemic is an epidemic on a worldwide scale. So COVID-19 would be a great example of a pandemic. So let's talk about the development of disease and the different periods of disease. So first you have incubation, which is the time between your infection and the appearance of signs and symptoms, okay? The prodromal period is next, where you have early, very mild symptoms, like you feel like you might be getting sick, that kind of thing. And then you have your period of illness, where it's the most severe. If the illness is not overcome in this period, the patient will die. If the patient doesn't die, you have a period of decline where the signs and symptoms are starting to reside. And then you have your period of convalescence or recovery. Interestingly enough, you are contagious throughout this entire process, okay? One of the most frequent ways diseases are spread are before people realize they're sick. Okay, before you start to feel symptoms, you may already be producing infectious particles. So etiology of disease, what causes disease? A lot of times this can be really challenging to figure out. Sometimes the signs and symptoms are non-specific, right? Sometimes we can nail the cause of a disease to a particular pathogen. There's a very famous scientist a couple hundred years ago named Koch who came up with 4 postulates to prove the cause of an infectious disease. His postulates are: it's the same pathogen present in every case, The pathogen is isolated from diseased hosts and grown in culture in a lab. That pathogen grown from culture in the lab can be put into a new animal and cause the same disease. And that pathogen can be re-isolated from the second animal. And it's the same pathogen, meaning that you know that that animal wasn't infected with something else. Same thing in a picture. So Koch's postulates don't always work. Some bacteria can't be cultured. If it's a virus, they only multiply inside living cells. So if you don't know you're looking for a virus and don't grow the host cells, you won't be able to grow the virus. Some diseases can be caused by many different microbes. Some microbes can cause many different diseases. And some pathogens only cause disease in humans. So working with them in a lab and in laboratory animals isn't going to help us at all. All right. So let's talk about our public health organizations. Who do we have today? We have the World Health Organization on the biggest scale. They monitor and report infectious diseases around the globe. They help develop and implement strategies for disease control and prevention, and they maintain a global alert and response system, giving logistical support and international response to epidemics and pandemics. In the United States, we have the Centers for Disease Control and Prevention, the CDC. The CDC's job is essentially to protect the public from disease and injury. A very much more difficult job than it seems. It oversees the National Notifiable Disease Surveillance System, which monitors outbreaks of diseases that could impact public health, like HIV, West Nile, whooping cough, Campylobacter infection, chlamydia, gonorrhea, COVID-19 now, those kinds of things. The CDC publishes a weekly publication called the Morbidity and Mortality Weekly Report, which gives physicians and healthcare workers updates on public health issues. And I gave you the link to it here if you're curious. In New Hampshire specifically, we have the Public Health Department of New Hampshire with the New Hampshire Department of Health and Human Services. They are responsible for the health, safety, and well-being of the residents of our state. This does include public health. So if you've ever watched the governor's updates on coronavirus, usually the head of the Department of Health and Human Services also speaks at those meetings. So a big part of what epidemiologists do is track data about infectious diseases. So it wasn't always easy to do. We didn't always have the technology we had today. We didn't always know where diseases were coming from. So a couple of hundred years ago, epidemiology was really started by physicians who were looking for patterns of disease occurrence and to understand ways to how to prevent it, right? One of the more famous early epidemiologists was John Snow. He worked in London in the cholera epidemic, and he looked at where patients lived who were infected and when, and he was able to trace it back to a single water source in London. In Florence Nightingale was a big proponent of hygiene and helped, she's considered one of the first epidemiologists because she really tracked diseases and their spread. Inga Semmelweis was a physician who really came up with the idea that hand washing can help prevent disease. This was before we knew where diseases were coming from. And then we have Louis Pasteur and Koch, who also helped. So all of these famous scientists a couple hundred years ago helped really develop epidemiology because we were learning a lot about where diseases were coming from at that time. All right, there are different types of spreads of disease. Okay, there's common source spread where there's a single source for all of the individuals that were infected, like a contaminated water source or a contaminated food source. There's point source spread where that common source operates for a short period of time. So like contaminated food at a picnic or a family gathering, right? And everyone gets food poisoning. There's intermittent common source spread where infections occur for a period, they stop and they begin again. Contaminated well water is a great example of that. So after a large rainfall, there's infection and that clears after a while. And then you have a heavy rainfall again and it comes back. So that's intermittent common source spread. And there's propagated spread. Propagated spread occurs through direct or indirect person-to-person spread with no single source for infection. Unless it's stopped immediately, infections occur for a lot longer than the incubation period. It can cause outbreaks that vary from small to large and can be very severe and not easily stopped. So what we're seeing right now is propagated spread, right? Indirect person-to-person spread with COVID-19. The types of epidemiological studies, the first one is observational, just data gathering, either by taking measurements or from interviews from people. The goal there is to make associations between disease and causative agents. Then we have descriptive epidemiology, which gathers information about an outbreak, the affected individuals, how it's spreading. That's the really kind of in-depth, okay, what is going on with this disease? How is it spreading? That kind of thing. Analytical epidemiology analyzes different hypotheses about causes for outbreaks. It can be retrospective, so looking back in time, or prospective as events unfold. And it can be experimental. Some epidemiologists will use labs or clinical settings to look at connections between diseases and causative agents or diseases and treatments. Reservoirs are organisms or non-living areas or sites where pathogens can persist over long periods of time. Non-living reservoirs are things like soil, dirty water. They might just naturally harbor the organism. Like the soil is a natural reservoir for tetanus bacteria. The environment could become contaminated with the disease organisms through feces or shed by hosts, or improperly treated wastewater is a big reason for fecal contamination in water, or even the remains of a deceased host can be reservoirs for disease. Sometimes pathogens can go into a dormant stage to survive, like endospores or protozoan cysts, if they are in a reservoir and not in a host. Some great examples are things like botulism, tetanus, cholera. Okay, humans, we are the biggest reservoir of human disease. We sometimes can carry and transmit pathogens without having symptoms. So if you're an asymptomatic carrier for coronavirus, you have the coronavirus infection, you're infected with it, your body will clear it, but you are, for that period of infection, you are making virus particles and you are contagious. That is an active carrier. If you're a passive carrier, you can transmit a disease mechanically. For example, if you're not properly washing your hands, and you're touching surfaces, or you touch something that's dirty and then you touch a surface that wasn't dirty to begin with, you transmitted that disease from one part to another. That's a passive carrier or mechanical transmission. Let's see. Animals, we talked a little bit about zoonotic diseases already. Animals are reservoirs as well, and they can transmit zoonotic diseases to humans either through direct or indirect contact. Sometimes the pathogen also infects the animal, like rabies, but sometimes it doesn't. Then we have our wonderful parasitic infections. They have a larval form and an adult form, so there are two different hosts usually, one that hosts the larval form and one that hosts the adult form. So the definitive host hosts essentially the adult form, the final form, definitive. It's the form in which the parasite reaches sexual maturity. Then the intermediate host hosts the other forms, the immature forms of the life cycle. All right, transmission. Transmission has to occur for infections to spread. It's that simple, right? Sometimes that's from a reservoir to an individual, or from an individual to other individuals, either directly or indirectly. So there's lots of types of transmission. The easiest one to understand probably is contact transmission, right? Literally physical contact, person to person spread. That can be vertical or horizontal. Vertical are mother to child during pregnancy, birth, or breastfeeding. Horizontal or between person to person. A lot of times that's between mucous membranes, skin to skin contact, or genital contact. Sometimes that can be droplet transmission. So contact transmission can include droplet transmission if that is a droplet that has traveled only a short distance from the host. So things that are coughed or sneezed or talked or even breathed out from one host to another in a radius of 1 meter or less, that's droplet transmission. If it's more than a meter, it's called airborne transmission, which is a type of vehicle transmission we'll talk about later. If it's a short distance in a droplet particle, that's droplet transmission. That's direct contact transmission. Indirect involves objects called fomites that have become contaminated by pathogens, either from an individual or from a reservoir. Examples of fomites are droplets from a sneeze landing on a tablecloth or a carpet and then picked up. Someone wipes their nose and then touches a doorknob or a towel. The doorknob is a fomite, the towel is a fomite. Items that are in clinical settings that are improperly cleaned or sterilized, things like tissues, diapers, linens, those kind of things. Anything that contains a pathogen that is not living can be an indirect mode of transmission in a fomite. Fomites are a huge cause of healthcare associated infections. A lot of times those fomites can be hands or surfaces. So vehicle transmission is different than direct transmission because pathogens are traveling through water or food or air. Water, that's pretty straightforward, right? Improperly cleaned water or improperly treated wastewater, right? Leads to contaminated drinking water, which leads to people that are sick. Contaminated food, that one's pretty self-explanatory as well, either through poor handling or storage or through cross-contamination. A great one that's relevant to everyone at home is preparing your meat and then preparing your non-meat food like vegetables. They tell you not to use the same surfaces for those, at least without cleaning them in between, because obviously the salmonella from your chicken will live on your cutting board until you clean it off, right? So if you put your vegetables on the cutting board after the chicken is gone but hasn't been cleaned, then the vegetables are going to pick up the salmonella, right? Great example there. And then there's airborne transmission, in which the pathogens are carried on dust or fine particles, including fine droplet particles that are traveling through the air, okay, for larger than 1 meter from the infected source. Then we have vector transmission. Vector transmission is a huge problem. It is essentially when insects like mosquitoes or ticks are carrying disease. If it's a mechanical vector, that means that the insect has picked up the pathogen on its feet, essentially, and has flown somewhere else and deposited the pathogen where it landed. If it's a biological vector transmission, that means that the insect, it has taken in the pathogen, usually by blood meal, biting a host, right, taking their blood, and then transmitted to a new host the same way, right? So like mosquitoes, or ticks and Lyme disease, right? Mites, lice, mammals, and rabies, or poultry and bird flu, those sorts of things. Here are a couple great pictures from your textbook, a mechanical vector versus a biological vector. Okay, and let's talk about prevention of transmission. Well, one of the first ways disease transmission was prevented, and still a common thing that's happening today, is quarantining, which we are now very familiar with. Quarantining is the process of isolating an infected individual or individuals suspected of being infected or exposed to prevent transmission of disease to others. Okay, so people infected with COVID-19, if they're not in the hospital, they're told to quarantine themselves at home, right? Isolate yourselves, stop the spread by not exposing yourself to other people, right? It's not always infallible, but it helps. A lot of people are self-quarantining right now because they don't want to get sick, right? So they're doing the opposite thing. They're staying home, staying away from pathogens or potential people who are infected, spreading pathogens, right? So there's both sides of that, but quarantining, as the term implies, is isolating infected individuals. We can even do this in hospitals, in isolation wards or special rooms that are either pressurized or negative pressure rooms that handle the air in very specific ways, that have like airlock chambers, or you have to gown in and gown out, those sorts of things. That is a way of quarantining in a hospital setting. There is a lot of transmission in places where there are many people housed. What we've seen with coronavirus was even on cruise ships, right, where you have a lot of people, they're in an isolated environment. They can't really separate from one another in that environment, and disease spreads rapidly through the population. Prisons, retirement homes, and nursing homes. Nursing homes are especially vulnerable because the population of people living in those nursing homes are vulnerable in general, right? They're there because they're aging or ailing and cannot take care of themselves. So in those locations, special measures have to be taken to limit the risks of infection. Then we have nosocomial or hospital-acquired infections. Right, you guys are probably pretty familiar with these and how they spread. The definition of a nosocomial infection is a disease that has been found in patients who did not have that disease when they were admitted to the hospital. Okay, and we think that they developed that infection while in the hospital. If you're in the hospital, you probably have a compromised immune system in general. Okay, so it's a very common spot for infections to develop. A lot of times those infections are infected with or connected with surgery or invasive procedures that give pathogens direct access into a host through contaminated surgical equipment or things like that. A lot of steps are taken to prevent infections at surgical sites, like use of antibiotics and things like that during surgery. But other nosocomial infections are things like pneumonia, urinary tract infections, infections that spread to the bloodstream and cause sepsis. Those are all very bad things that can happen, right? Let's talk a little bit about MRSA. I know I've mentioned MRSA before, but it's Staphylococcus aureus that has become resistant to methicillin, so methicillin-resistant Staph aureus. Originally, Staph aureus, it lives on the skin. It can cause a wide range of human infections. In the 1950s, penicillin was developed, and so they started treating that with penicillin. Well, Staph aureus very quickly evolved resistance to penicillin, so they started treating it with methicillin. And then in the 1980s, resistance to methicillin started arising, so that's where the term MRSA came from. And now, if they encounter Staph aureus that's resistant to methicillin, they treat it with vancomycin, and now we're seeing vancomycin-resistant Staph aureus pop up. So there are essentially every different antibiotic we throw at this bacteria, it's evolving resistance to. So it's definitely a very potentially bad pathogen, right? And then we have our emerging and re-emerging infectious diseases. A disease is emerging if it is either new to the human population or showing a very large and quick increase in prevalence, or an increase in frequency after a previous decline. So examples of emerging or re-emerging infectious diseases are things like COVID-19, Ebola, measles is coming back, all of those things that are coming up from either nowhere or from very small amounts, right? And so our public health facilities, the WHO, the CDC, the National Institutes of Health, and the National Institute of Allergy and Infectious Diseases, those are two government organizations that do a lot of research and grant giving for other research scientists. They monitor and help with outbreaks as well. So some contributing factors for emerging infectious diseases, either new strains that arise from genetic combination or zoonotic spread. They can come from resistant strains arising from use of antibiotics and pesticides, changing weather patterns. Global warming means more mosquitoes, means more spread of mosquito-based diseases. Transportation, you can have Ebola and not know it and get on an airplane and fly somewhere else in the world and take that Ebola with you, right? We saw that firsthand a couple of years ago, and thankfully we were able to control that very quickly. But it did happen, and that's how COVID-19 is spreading, through spread of people, right? Ecological disaster, war, expanding human settlements, deforesting the rainforest, all of those things are exposing us to diseases that were previously kept in their original environment. Animal control, so killing of predators or vectors, and the changes in the food chain that causes can cause different diseases to emerge, and failure to vaccinate, which is honestly a public health failure. Not vaccinating is causing the emergence of things like measles that used to not be a problem. So let's talk about vaccines while we're on that topic. The goal of vaccination is to protect an entire population against disease, okay? Because immune individuals will prevent the spread of disease. okay? So the goal of vaccines is to stop the spread of diseases through populations. In order for that to happen, you need to have most of the population be immune to that disease. So you need to have most of the population be vaccinated against that disease, okay? Vaccines are relatively safe, right? The amount of adverse effects that happen from vaccines are very, very, very low statistically, okay? And the benefits largely outweigh the risks for most people, not for all people, but for most people, okay? So to make a vaccine, you take a pathogen or a piece of pathogen or a toxin made by a pathogen, and you make it so that it cannot hurt someone. You inactivate it or weaken it to the point where it can't hurt someone. And then you add some other molecules that kind of beef up the immune response and inject that into someone. And the body sees that as a pathogen and it mounts a response to that pathogen, right? It creates antibodies or T cells against that pathogen. And then it remembers that pathogen. Okay, we'll talk about immunology in a few lectures, but after the immune system is exposed to a pathogen, it remembers that pathogen. And if that exposure is strong enough, it will be able to reactivate quickly against that pathogen if it's seen again, and to stop the disease before it causes infection in the host. So we talked about morbidity versus mortality, prevalence and incidence, patterns of disease, periods of disease, etiology and Koch's postulates, the organizations that help us track disease, the pioneers in epidemiology, which I have videos on for you in Canvas, the types of spread, the types of studies that epidemiologists do, reservoirs, types of transmission, nosocomial infections, emerging infectious diseases, and vaccines. All of these things are incredibly relevant to our day-to-day lives. And so I hope that you were able to take something from this lecture that was meaningful for you. Thank you.